Hey there, I'm Anna, and today we're taking a trip through time. Okay, so I don't exactly know how to build a time machine. But with techniques in DNA sequencing, scientists are learning to investigate the past and identify the species of the future without needing a time machine. They can study the evolution and classification of organisms using just a tiny fragment of genetic information. To understand these techniques, we first need to remember how DNA itself functions. DNA consists of long strands of nucleotides, which form segments called genes. Within a cell, DNA is transcribed into RNA and then translated into long stretches of amino acids, which can be thought of a bit like beads on a string. The order of these amino acids determines which proteins they fold into, and different proteins cause different traits within organisms. Over time, mutations, subtle changes in the original DNA, occur randomly, causing differences in the final proteins and traits that are formed and leading to variation within and between species. Since the 1950s, scientists have theorized that looking at the differences between a sequence of amino acids in two organisms could allow us to determine the amount of time that has passed since those species shared a common ancestor. This idea is known as an organism's molecular clock, or the average rate at which it has changed over time. It has fascinating potential for our understanding of evolution, because unlike fossils, which are rare, thousands of proteins can be found in almost every living creature. If we can determine an organism's history by looking at the proteins it has today, it means we have access to tons of potential information. The comparison of this information forms the basis for another technique, DNA barcoding, in which scientists isolate a specific sequence of DNA and compare its makeup in different organisms. In animal cells, a gene known as cytochrome C oxidase 1 is perfect for this because it mutates much more quickly than other sections of DNA, so the order of nucleotides found in this part of a cell differs significantly between species, even in those that look a lot alike. Just like barcodes on clothing or food are used to store information about these products, DNA barcodes store genetic information, which can be used to tell species apart. The CO1 genes for thousands of species have been sequenced and added to reference libraries, which track the particular order of nucleotides in the DNA of registered species. When an unknown organism is discovered, its DNA can be compared to previously identified organisms using these databases. As technology improves, we'll be able to sequence smaller and smaller sections of DNA, allowing us to produce more accurate molecular clocks that deepen our knowledge of the history of life on Earth, and create more specific DNA barcodes that reveal hidden differences between species.